Okay, everybody. So let's carry on with our discussion about DNA function. We had described exactly how the replication of DNA takes place, and that was easily done because each strand of DNA codes for the other. So we can separate them and easily replace the bases on either to create two new DNA strands. So that property depends upon the ability of bases to pair with one another. Base pairing rules are extremely strict. A always bonds to T, C always bonds to G. And that means if we have a single strand, we can put matching bases together with the other bases in the strand. The same principle is going to be applied now in the actual functioning of DNA in actually producing proteins from the DNA code. The DNA base code is lodged in the sequence of bases on one of the strands of the DNA in any gene. And for the most part, a single gene codes for a protein. There are some exceptions that I will mention in a minute. But locked into the DNA sequence of bases, there is a code, and the code is for amino acids. So somehow we have to take this molecule, which is the nucleic acid, DNA, a sequence of nucleotides. Nucleotides are the monomers of DNA and of the nucleic acids in general. We have to take that code and we now have to change it into a sequence of amino acids. Amino acids in sequence form a polypeptide, which goes on to form a protein. So somehow we have to be able to take a code in nucleotides and translate it eventually into a sequence of amino acids. And that sequence is extremely specific, and it is determined by the sequence of bases in the DNA. But to move from DNA to the polypeptide, that requires several rather complex steps. And all the way along in that process, the intermediary molecule is another nucleic acid, which is RNA, ribonucleic acid. Very, very similar to DNA in that it is composed of four bases, four nucleotides, rather, with their attendant nitrogenous base. So the differences between DNA and RNA are, first of all, that ribonucleic acid is fundamentally single-stranded and not double-stranded the way that DNA is. However, even though it is single-stranded, different regions of a single RNA molecule can double back and can actually base pair with its own bases, its own base sequence somewhere else on the RNA molecule. And this enables the RNA to form complex three-dimensional structures in space. So we find RNA in many different roles, and amongst them is that they can form structure. They can be structural RNAs. So but they remain fundamentally single-stranded. We'll see examples of this in a while. The second difference is, although they have four nucleotides, the nucleotides all have basically the same structure as those in DNA, with one important difference, and that is that at the 2' carbon, go back and remind yourselves about how the carbons are numbered on the molecule. The 2' carbon, they carry an OH group, a hydroxyl group, not an H. In DNA, there is only an H at the 2'. That is why it is called deoxyribonucleic acid. It's been deoxygenated at 2'. But in RNA, the oxygen is still there in the OH, in the hydroxyl group. The third important difference is that RNA does not use the base thymine here. Instead, it uses a base called uracil. And uracil is extremely similar to to thymine. But it is a way for the cell to recognize that it is dealing with an RNA molecule, not a DNA molecule. It's a label, in other words. So those are the three important differences. So we're going to also encounter different kinds of RNA. There are many kinds of RNA inside the cell. And there are many functions to these different RNA molecules. But for the moment, we're only going to be dealing with three of the important ones. And these are, first of all, messenger RNA. A messenger RNA is a direct transcript from the DNA, from the strand of the gene which contains the genetic information. There are two strands in the gene. Only one of those strands contains the sequence for amino acids. That strand is called the template strand. Rather confusingly, the other strand is called the coding strand. What does it code for? It codes for the template strand. It's the matching DNA strand. The template strand is the strand which contains the information for a sequence of amino acids. The other strand codes for the template strand so that they are matched, and of course, by base pairing. Messenger RNA is read off the template strand. It is produced from the template strand of the gene. Now, when you look at the whole DNA molecule, you'll find that in one gene, one strand may be the template strand. Go to another gene, and you'll find that the other strand is the template strand. It just depends on the gene. It depends on the gene as to which strand is acting as the template strand. So the messenger RNA is tremendously important because it is literally the message. It's the message from the DNA to the rest of the cell. It is carrying the information, a direct transcript of DNA information to the rest of the cell so that that message can be translated, and it can be translated into proteins. We got a sequence of DNA bases, DNA nucleotides, that is transcribed into a messenger RNA transcript, and then the cell is going to translate that messenger RNA transcript. So the next species of RNA is the RNA that actually performs this translation between the pure nucleic acid of DNA and messenger RNA into an amino acid sequence. And this is this molecule called transfer RNA. And transfer RNA is where in the cytoplasm, if it is charged, we say it's charged when it carries an amino acid. And there are many species of transfer RNA because any one transfer RNA can only carry one specific amino acid. We use, living things use 20 different amino acids with a few bizarre exceptions. But in general, you can assume that living things use 20 amino acids. And so we've got to have at least 20 different transfer RNAs in order to cope with the 20 different amino acids that they're going to have to carry. In fact, there are more. There are more transfer RNAs than that, as we'll see in a while. So the transfer RNA literally transfers in two ways. It transfers the information from messenger RNA to the ribosomes. And not only that, it physically transfers amino acids. So it's doing two things. It's translating the messenger RNA code, and it's carrying the amino acids to form the amino acid sequence eventually. The third type of RNA that we're going to encounter is ribosomal RNA and rRNA. Ribosomal RNA has two important functions. The first is the ribosomal RNA is often very, very large RNA molecules that fold themselves into very complex shapes, which form the structure of the ribosomes, hence the name ribosomal RNA. There are several kinds of ribosomal RNA. A ribosomal RNA are a little bit different in prokaryotes to what they are in eukaryotes, but the structure and function is essentially the same. The body of the ribosomes, which are going to perform the act of translation by interacting with transfer RNA and the messenger RNA, the ribosomes themselves are made up of a mixture of ribosomal RNA and protein. But here is one instance where the ribosomal RNA, has enzymatic ability. It is able to act as an enzyme, and we refer to it as a ribozyme. Remember, I told you much earlier in the class that the vast majority of catalytic ability, of enzymatic activity inside the cell is conducted by proteins. But here is an exception. And this is the action of ribosomal RNA as an enzyme. We'll refer to it again in a while. I just want to show you the structure, a little bit about the structure of transfer RNA. And we'll refer to this again later on. But I wanted to show you that transfer RNA is actually a relatively small molecule. But here's an instance where we can see that a single-stranded RNA molecule can actually form these very, very complex structures. Here, for example, this here, up here, this is the 3' end there. Follow the molecule, and you'll see all the way around is fundamentally single-stranded. But look here, it is base pairing here to its own. That part, that 5' end, is base pairing towards this 3', this side. Where this occurs, where base pairing like this occurs, just as in DNA, it will form a helix, a double helix in space. It'll twist the molecule around. There are areas which are unpaired, which buckle out into these loops here. And remember, all of this, now this is kind of flattened just for convenience sake into 2 dimensions. But when we look at it when we model it in 3 dimensions, it assumes this sort of upside down L shape. And here's the 3' in here. Trace it all around, and you can see these little helices here. You can see the loops here. And all of this twists the RNA molecule into this very complex 3-dimensional shape. I will just mention now, but we'll talk about it in a little bit more detail in a minute. There are 2 really critically important areas of this little molecule. The first is the 3' end here. It's called the acceptor stem because this is where an amino acid binds there. And the other important end is here. There are 3 bases here whose code is for this amino acid. So in other words, each transfer RNA molecule, it has a little code here at that end. It's actually called the anticodon for reasons you'll see. It has this little area here of 3 bases that match exactly to the amino acid that it is carrying here. We'll discuss it in more detail in a minute. Okay, so putting these all together, what we come up with is what is referred to as the central dogma. And the central dogma because it applies to all living things. Everything that is alive uses DNA as the basis for the functioning of the cell. because the DNA codes for everything that the cell needs to function, to perform the functions of life. But the DNA doesn't operate directly. Instead, it is a code largely for proteins, enzymes that will drive the functioning of the cell. So we have to get from DNA to the polypeptides, which will eventually make up those functional proteins. The central dogma refers directly to the way in which that's done in all cells. And in all cells, DNA here is transcribed to messenger RNA here. And that messenger RNA has to be translated. And it is translated to protein, polypeptide, by the ribosomes there. So that is the central dogma. And this is something you need to memorize. You need to have it absolutely clear in your head, because this is one of the most important findings in all of biology, is the DNA is transcribed to messenger RNA, which is translated to protein by the ribosomes. So what this is going to do is it's going to take genetic information lodged in the DNA, and it's going to make it functional inside the cell. All of that requires these intermediary molecules: messenger RNA, transfer RNA, which is going to bring in amino acids and is going to eventually end up producing the protein, polypeptide, which will form the protein. You should phrase it that way. So the first step is what we're going to discuss today, and that is this process of transcription. Transcription is the production of a messenger RNA strand from the template strand of the gene. And the step here. So the DNA template is read from 3' end to 5' end. Why? Why is it read from 3' end to 5' end? Think for a minute. It has to be read that way because we can only make the RNA strand from 5' to 3' direction. You can only build the RNA strand by adding nucleotides at the 3' end. Okay. And so if the RNA strand is being made from 5' to 3', it must be read off 3' in the 3' to 5' direction of the DNA. There are complex ways in which this transcription is controlled. There are certain genes that are read off the whole time, that are transcribed the whole time, and those are called the housekeeping genes. They are producing things which are continually used and need to continually be replaced, or in some cases amplified inside the cell. So, for example, proteins involved with cellular respiration, those are used up the whole time, and break down and have to be replaced. Those are read off the whole time. But many other genes remain silent until they are needed. It would be chaotic and a waste of energy if all genes were being read off the whole time. Instead, very careful control has to be placed on the genes, on the usage of those genes. And the control is very often at the level of transcription. We control the transcription of the gene, and then we control how much of the gene product is going to be produced. So there are various ways in which this takes place, but the simplest way to think about it is this. A gene doesn't only consist of that area which is transcribed and going to eventually produce the protein. The gene may contain a lot of other information. And a lot of that, the control information lies, we say, upstream. Transcription is going to begin at a certain place on the DNA template strand. But upstream, above there may be information which controls the process of transcription. So, for example, above where the transcription will actually start, there's an area called the promoter sequence. Promoters are very complex. We're not going to discuss all of the details. We'll hear a little bit more in a while. But it is in that promoter area that effectively a switch is placed to ensure that the gene is only read off when it is actually necessary. So once transcription begins, then we start building a messenger RNA strand, and the messenger RNA strand has to end somewhere. There are various ways that this can happen, but effectively, there is a terminator area, a terminator sequence somewhere there, which does various things, which may have various structures, but its effect is to stop the process of transcription, so that we end up with a certain length of messenger RNA, which then has to be exported into the cytoplasm for the process of translation to begin. So here's the process of transcription. And if you understand DNA replication, it's relatively easy to understand the process of transcription, with the important difference, of course, that transcription is only of one strand of the DNA, and that is the template strand. So just follow here, and you'll see this is our template strand there. And it is the messenger RNA is going to be produced by a set of enzymes which together form this whole complex here, the most important of which is RNA polymerase. RNA polymerase, because it's producing an RNA strand. We heard before about 2 species of DNA polymerase, but here there's only one RNA polymerase that we're going to talk about. And that is going to produce the RNA, the messenger RNA strand from the template of the DNA. So the first thing to realize is, here's what I meant by saying that the promoter lay upstream. Here's my template strand, and the template strand is going to be reading 3' there. Sorry, 3' here to 5' there. Okay, it's going to be reading 3' to 5'. And the reason for that, we can only build the RNA strand at the 3' end there. So here's the RNA strand that is going to be produced. There's its 5' end there. There's its 3' end there. If I was to cut this DNA here, that would be a 5'. This is 3'. We're going to add on RNA bases here to make the messenger RNA transcript. So this is what the RNA polymerase does. It separates, or the RNA complex, I'll say that. It's, first of all, binds upstream here. That is in the promoter area. There are various controls over its binding in that region, which we're not going to discuss in detail. But once it is firmly bound, it moves down this promoter region of the transcript, of the template rather. It moves down until it reaches a point where it is actually instructed, begin transcription, begin producing the messenger RNA transcript, so many bases down this template. So it binds here, reaches a signal that says, okay, in 20 bases, start the transcript. That's effectively what happens. And it will begin to lay down RNA bases which match the DNA bases on the template. Look at how that base pairing occurs. It is the usual base pairing, C to G, G to C, as you see here, A to T, where A here is on the RNA, is binding to, is base pairing with T here on the DNA. But look what happens when we have an A on the template strand. RNA does not use thymine, it uses uracil. So we have here A to U. Okay. This RNA transcript is growing in this direction here. The whole polymerase complex moves in this direction there, that way. It's adding bases at the 3' end of the RNA transcript here. Eventually, the RNA polymerase reaches an area which terminates transcription. As I said, there are various ways in which that signal can be given to the RNA polymerase, but its effect is always that the RNA polymerase leaves the DNA, divorces from the template, in other words, and releases the messenger RNA strand. It's a relatively simple process to think about. It's a relatively slow process compared to the replication. It's not necessary for this to be done extremely quickly, like the way that transcription has to be done. This is the function of the gene. It doesn't stop the functioning of genes the way that replication does. Okay, so having a look now at putting that all together with the linear structure, let's say, like put it that way, of the gene. Here is an area which comprises a gene on the DNA. The promoter is this whole area upstream of the area that's going to be transcribed. The promoter binds here, and the RNA polymerase moves down a certain specific number of bases. When it reaches the start area here, it will open up the DNA and it will begin to transcribe. This here is referred to as the transcription bubble, and it's relatively short. Maybe less than 100 bases that is opened. It moves down, the polymerase moves down, and all the while it is transcribing, and so a messenger RNA strand begins to trail behind the polymerase. Eventually, the RNA polymerase reaches the area which is a terminator. At the terminator, the RNA polymerase leaves the DNA template, and it releases the messenger RNA. Now, the fate of that messenger RNA differs whether we're talking about a eukaryote system or a prokaryote system. In the prokaryotes, the messenger RNA can be used as soon as it appears in the cytoplasm. That means, in actual fact, that ribosomes can begin to bind to it even before the transcript is complete. Because in the prokaryote, this is trailing off, this transcript here is trailing off into the cytoplasm. Ribosomes can bind here and begin the process of translation even there. But in eukaryotes, a different system has to be adopted because the DNA is all contained in the nucleus. And the nucleus does not contain ribosomes that are able to translate the transcripts that are produced. Instead, in eukaryotes, the messenger RNA transcript has to be exported from the nucleus. It has to be exported through the nuclear membrane into the cytoplasm before anything can happen. In eukaryotes, in addition, there are various other things that need to happen. The RNA actually is processed. In prokaryotes, that is not the case. As the transcript comes off here in prokaryotes, it's usable. No further processing needs to take place. Okay, so I'm not going to discuss all of translation today. I'd rather do that in person. But I am going to just introduce you to the first ideas about how translation takes place. So what have we covered for our DNA, for the central dogma? DNA is transcribed to messenger RNA, which is then translated to protein by the ribosomes. We're halfway through the process, in actual fact. But the process of translation is a little more complex. Transcription is easy to understand, especially if you understand replication, because it's rather similar, excepting it's producing a messenger RNA transcript, not a new DNA molecule. So what we have to do, you have to realize messenger RNA is a direct transcript. It's essentially a copy. It's like of the DNA, the genetic information in the DNA, of the DNA sequence. But it's an inverse copy. If you were to look at it, if you were to look at the transcript, you'll see it actually bears a very, very strong resemblance to the coding strand of the DNA, although it's a transcript of the template strand. Here's the key to the whole process. When we look at that messenger RNA transcript, we'll see that the transcript is grouped into words, and each word is 3 letters long. Those 3 letters are either C, U, G, or A. The letters are made up of the 4 bases of RNA. They're grouped into groups of 3. Each group of 3 is called a codon, and each codon is specific for something, usually an amino acid, sometimes for an action, sometimes for an action and an amino acid. It'll become clear in a minute. If I look at the math of it, I've got 4 letters and I've got words made of 3. That means I can make up 64 words. 64 words can be made from those 4 letters. And those are the codons. Those words are called the codons. And 61 of the 64 code for amino acids. So this code that we've got locked into DNA and then into the RNA. In the RNA, we've got 61 codons, which we call the sense codons, which encode amino acids. We have 3 codons, which code for an action only. And so we refer to this as degenerate or redundant code, because it means we've only got 20 amino acids and we've got 61 codons that could code for them. So for some amino acids, there are several different codons. For others, there's only one. But for many of the amino acids, there are several different codons that can code for them, and they all basically operate the same way. And the other important thing to realize is that for those 61 codons, each one has a specific transfer RNA. And transfer RNA is linked to the codon and to the amino acid that the transfer RNA is going to carry. So let's have a look here. Actually, I'll just do this for a start, but I've got a rather easier table for you to look at. And So here is a list of all of the codons. And you can count through, and there are 64 of them. And the codons, obviously, they consist of 3 bases in sequence. So, for example, if I have a codon that reads U, it codes for the amino acid phenylalanine. And let me go here because I thought I had one. Oh, okay. I thought I had one with complete letters, so it doesn't matter. So you can, if you are trying to translate for yourself, you're given a codon and asked, what is this code for? You can chicken peck your way through everything and find it if you want to. But that's actually not necessary because the table of codons is actually arranged so that it's easy to find any particular codon. The first thing I need to emphasize is this here is the 5' end of the codon. So this is 5', and this is the 3' end there. So this reads 5' U, 3'. Okay. So here's the first thing to notice. There are 61 of these codons that code for specific amino acids. But 3, that leaves us with 3. Those 3 code for an action. And that is that they are called the stop codons. And when they occur in a messenger RNA sequence, they signal to the ribosome that the process of translation is to stop and the whole process ends. So these 3 stop codons are UAA, UAG, and UGA there. There is also a start codon. There's a codon which instructs the ribosomes start translation here, and it is here, and that is AUG 5', AUG 3'. And this is also a codon for methionine. So this is one of them that you are to remember. 5' AUG is the codon for methionine. There's one other thing I want to point out, and that is that these codons originated in a DNA code. So if let's just go here, what is the DNA code that produced this 5' U? It is 3' A. What about our start codon? What is the DNA code sequence that led to 5' AUG? It was 3' T A C. Write it out and you'll understand. That 3 nucleotide sequence in the DNA is called a triplet. It is the DNA triplet. A DNA triplet produces a codon in the transcript. So AAA produced UUU in the transcript, in the messenger RNA transcript. You'll see also that there are some of the amino acids have lots of different codons that code for them. Here's serine, UCU, UCC, UCA, and UCG. These all code for serine, all 4 of them. And there are others with even more. Some of them just have relatively few. Here you see phenylalanine only has these 2 here. Leucine has 6. 1, 2, 3, 4, 5, 6. And so that is what we mean by saying that this code is degenerate or redundant. A single amino acid may have many different codons that code for it. So that is fine. We will actually do various exercises, seeing how we derive different amino acid sequences from both the messenger RNA and from the DNA itself. That is a classic kind of exercise to do, is not only to test you, but actually will help you to understand how these processes take place. I do not expect you to remember the names of all of these amino acids. Remember, though, that there are 20 of them. The one you must remember is this one, methionine, which is the start codon. And the 3 stop codons, UAA, UAG, and UGA. I will actually give you another table of codons with the amino acid names written out in full. I thought my next diagram had that, but it doesn't. I'll make sure that you get one. Okay, and you're going to be presented with several different exercises using the table of codons. So there is this, I told you that the table is organized so that it's relatively easy to find a particular a particular codon with its attached amino acid. And this is the way it works. Remembering this is 5' this side here. Here's an animation for you. And I'll just explain that the first, you see here, this is the first amino acid in the codon sequence. So if I tell you, let's look up CAG. So what's our first codon, our first nucleotide? Cytosine C, right? Here's the second base here. So we've got C. Well, it has to be in this column, this row here. So go C somewhere here. The second one was A, so it has to be here. And well, you can probably spot it already. But nonetheless, here's the third base. And we go third here, CAG. And that's for an amino acid called glycine. So that is, and that's the way it works. It's really quite simple.
The 4 nucleotides.
Okay, so we're going to stop there because we're going to go, when I see you on Monday, we'll discuss the process of translation itself, which is the responsibility of the ribosomes interacting with both messenger RNA and with transfer RNA. And we'll see exactly how that's done on Monday. Okay,